we're very pleased to continue with the next segment of the program. I think that although we're very much focused on how to handle the COVID-19 pandemic, these are extremely exciting times in the management of myeloma, in the research of myeloma, and particularly the evolving role of the immune therapies, treatments that work with your own immune system, and as we touched on earlier, CAR T-cells and bispecifics, really exciting area of research and treatment. And so it's just fantastic that Paul Richardson is available and able to be with us today. And so please, Dr. Paul Richardson from the Dana-Farber in Boston, very grateful for your presentation today.
Great. Thanks very much. Hello?
There you
are. Hi, Brian, Susie. A very good morning in LA, and actually still morning here in Boston, actually. It's lovely to be with everyone. It's one of the highlights of the year, and I certainly will not forget last year when we presented just as the pandemic began. And really to echo something that Donna so nicely said, I think we do see true light at the end of the tunnel, but like all of these things, it's a marathon, not a sprint. And I think for us as myeloma caregivers and patients, we can certainly relate to that. Having said that, to echo your point, Brian, the optimism in myeloma is real because despite the enormous challenge of the pandemic, progress has been remarkable. And in that regard, what I wanted to do today, really on behalf of colleagues who've really led the field in immunotherapy, is frame that for our listeners and at the same time discuss with you some of the innovative immune therapy that may not be quite so obvious, such as the impact of cellular modifiers, the so-called cell mods. I'm going to touch on those as well. So in that spirit, these are just my relevant disclosures. So as we think about key targets in myeloma, I think it's very important to help frame the conversation by addressing the issue that this disease is incredibly genomically unstable as an entity, and therefore the targeting and overcoming of mutations is a primary focus in myeloma research. And in that sense, there is a critical role of combination and continuous therapy. And Brian, I thought you framed it beautifully earlier. You know, for example, the regimen of RVD is a paradigm from the beginning of development, which obviously we led in 2006 to your pivotal trial that followed. We've seen a remarkable outcome from the 3-drug platform of proteasome inhibition, immunomodulation, and steroids. And now, of course, with the RVD-DARA paradigm moving forward as an example, and of course also the success of KRD, we've realized that this combinatorial strategy plus an antibody is changing people's lives and dramatically improving prognosis. And I was particularly impressed to see the long-term follow-up of SWOG 777, very similar to the long-term outcomes that we've seen from our original early phase studies over 10 years ago. So again, really important hope for our patients. But beyond that, the challenge then becomes, well, where do we go? And I think it's important to recognize that there's an evolving position and timing of transplant, which I think is very exciting because obviously we're learning very quickly that one size does not fit all when it comes to transplantation, and that high-dose melphalan, great as it's been for many, many years, the position of that particular approach is changing with the success of these other combination strategies and the fact that we can offer now to our patients the delayed option of keeping transplant in reserve and using it when necessary, as opposed to, you know, in a more algorithmic fashion that could be considered now perhaps somewhat outmoded. So with that in mind, what are the sort of new directions that we're heading in? Well, obviously a cornerstone of what we've been doing is targeting excess protein production, all the forms that that takes, be it with proteasome inhibition or, for that matter, in the context of protein degradation strategies and others. But critically, the restoration of immune function in myeloma patients has been recognized to be fundamental. And immune suppression in myeloma has been a frontier from the very beginning, obviously. But now we have real strategies that can be targeted at overcoming it. Now, why this is so important is summarized in this slide. I'm very grateful to my partner, Nikhil Munshi, for sharing this with me. This is a whole genome sequencing profile on one of our patients who undergoes initial diagnosis. This is on the left of the slide. You can see complex genomic changes at diagnosis, and this patient actually has over 5,000 mutations in the genetics of their cancer at diagnosis. This patient then undergoes induction remission therapy with RVD, as it happens, and then followed by high-dose melphalan, further treatment, and then maintenance, and then unfortunately relapses. And at the end of that point, when we looked at the whole genome sequencing here, you can see the number of mutations dramatically increases to over 12,000. And this gives you one arrow. This is from Nikhil, showing one targetable mutation in this particular case. But I show this because you can see the challenge is, well, that's great that that's targetable, but look at the rest of this. You know, we're dealing with 12,000 mutations strong in this patient's disease. How on earth do we shut this process down? And so as we think about how our drugs work and think about how we throw a big net around this, and we'll hear in a minute from Rafat in more detail about strategies in relapsed refractory disease and why this so matters, I think it's very obvious from this picture, it sort of says 1,000 words in my view, that strategies such as immune treatments that can grapple with the wholeness of this problem and shut it down, clearly intuitively make great sense. Now, I'm just going to, oops, sorry, slides are jumping a little bit. This is actually just an algorithm that I just want to share with you, which I think sort of illustrates the point I'm trying to make. We should think about immune therapies in two ways, active and then arguably sort of passive. And I would say the active strategies are those designed to act on the immune system itself, and the passive ones are those designed to act directly on the tumor. Now, we'll hear in a minute from Rafat about anti-tumor monoclonal antibodies. He's also going to talk about antibody drug conjugates. I'm going to touch on those as well, simply mechanistically, because I think they're a very important strategy. And also, of course, I've already mentioned the immunomodulatory drugs. We'll go a little deeper into that with the discussion on the cell mods. But of course, my focus in the interest of time today for everyone is really on adoptive therapies, cell therapies and adoptive T-cell therapy in particular. But I would be remiss if I didn't mention, obviously, the importance of vaccines, which we're in the process of developing, especially in the smoldering setting or early stage setting, where I think they have potential real opportunity as well, arguably in the post-CAR T setting of re-educating the immune system. And after all, we've been talking all morning about the importance of vaccines in COVID. And needless to say, one of perhaps the gains of the COVID tragedy and all that's gone with it, though, is our ability to really think hard about the vaccines that have come out from this extraordinary response to the COVID disaster, and then be in a position to think, well, maybe some of the lessons we're learning there could come across nicely into myeloma. But the good news is we already have vaccine strategies well underway. Now, just because before we get too fizzy about immune therapy, it's worth mentioning the cautionary tale of the checkpoint inhibitors, where checkpoint inhibitors initially showed great promise, but actually is good, and the complexity of this process became very clear. So I think as we think about immune therapy, it's important to bear in mind that this is an incredibly fast evolving field. But the good news is there are very promising strategies, but also to remember that some things have been more challenging than others. Now, in terms of immune surveillance in multiple myeloma, we know this really matters. We know that there are multiple pathways through which these work. And again, I just show these by illustration, and they'll be available to all our listeners to review at their leisure in more detail. But I just want to emphasize the point that you can restore host immune surveillance, which is essentially the platform of the CAR Ts I'm going to come to. I want to emphasize the immunologic effects of myeloma therapies, and we'll touch on those in particular in the context of cell mods, but it's important to realize immunomodulators do that already. We've already discussed the vaccines, and then, of course, there's the strategies of antibodies, which Rafat is going to spend some more time on in a moment as well. Now, in terms of adoptive cellular therapies, a couple of points to make here, and I just want to acknowledge my colleague, Dr. Adam Sperling, who very kindly shared this particular slide with me, because I think it's very clear-cut and very, very, very informative that when we think of adoptive cellular therapy, what we're really looking to try and do is re-educate a patient's immune system, and in particular their T-cells, to then target disease accordingly. And CAR T, of course, has been a paradigm of this in a highly sophisticated fashion through T-cell collection, T-cell transfection, in which basically the antigens that we need to target are then integrated into these T cells. These T cells then are transferred back to the patient, and in anticipation of that, lymphodepletion may or may not be necessary. And then after that, there is a highly sophisticated approach to patient monitoring, both in terms of disease response and CAR T persistence, as well as management of side effects. So this, in a nutshell, is the construct between how the CAR T process occurs. Now, I want to, again, not to blind people with the science, but simply to illustrate the sophistication of this approach. Chimeric antigen receptor T cells, or CAR T cells for short, reflect both an extracellular domain, a transmembrane domain, and an intracellular domain that applies to how to manipulate, in particular, the T cells involved. And there are first, second, and third generation CARs. Now, again, not again to make this unduly complicated, but just to understand that this is a rapidly evolving space where essentially in the first generation CARs, we looked at what we would call T-cell co-receptors and T-cell activation being key. The second generation receptors then went a little further by generating what we call a co-stimulatory domain. And the third generation now are so exciting because they include two co-stimulatory domains. So the rationale is to yet further enhance the ability of the immune response to go after the patient's particular disease. Now, the target antigens in multiple myeloma are summarized here. And please, again, don't necessarily worry too much about the details, but simply just know that we have several: CD38, CD138, CD19, the kappa light chain, SLAMF7, BCMA, and a very exciting new one, GPRC5D. And clinical trials in all of this are now underway. The important message here is that whilst I'm going to focus, for the interest of time today, on BCMA primarily, to know that a variety of these targets are being pursued broadly. I think we're all very familiar with CD38, and again, Rafat will spend time on that in a moment. Perhaps people are a little bit less familiar with CD138, but interestingly enough, CD19 was one of the first targets exploited in myeloma patients. We'll come to that in just a moment, but most of the work to date has been done in BCMA. Now, in terms of CD19, this is actually illustrative of a patient who actually we cared for at Dana-Farber as well as at UPenn, patient of actually Dr. Anderson's. And she had a history that's summarized here, as you can see, relatively classical in the sense that she underwent autologous transplant, lenalidomide, bortezomib, and dexamethasone, so RVD-based therapy thereafter. She also had other combination novel treatments at the time, and then ultimately was effectively cytoreduced at UPenn with a second intensification and went on to receive a CD19 targeting CAR. Wonderfully, she had a great response, and actually this was very exciting to see. It didn't last terribly long, unfortunately, about a year, but then when the disease came back, we were able to control it much better because by that time we had daratumumab and other strategies, and she did much better in the long term and continues to do so. The important point, though, is this was our first excursion into CAR T therapy in myeloma, and whilst promising, CD19 actually did not translate for the other patients as particularly beneficial. So BCMA was the next on the block, and this has obviously been much more exciting, and we'll go into much more detail now. BCMA is a so-called member of the TNF receptor superfamily. I think it's best just to remember it as a B-cell maturation antigen. And the good news is that its expression is largely restricted to all plasma cells and some mature B cells, which provides a degree of selectivity. And what you can see on the right of this slide is the brown staining is the BCMA protein in a mass of plasma cells. And so this provides us with an ability to go very directly after BCMA. Now, the best platform for this so far has been the BB2121 platform, so-called ide-cel. It's the most advanced and hopefully will be FDA approved as soon as the end of this month. This takes an autologous CAR T-cell, and then with a co-stimulatory domain, as well as the intracellular signaling domain, has been able to be successfully targeted to patients with relapsed refractory myeloma. It shows the phase 1 design, again, not to belabor this, but just to acknowledge the tremendous work from investigators in this field, led by my colleague, Dr. Nupa Rajay. And this paper was published by Nupa and her team, including colleagues from our own center, and particularly my partner, Dr. Nikhil Munshi, in the New England Journal a couple of years ago. And in fact, we saw very promising results from this at the time. And Nikhil just published, in fact, about 10 days ago in the New England Journal, the phase 2 experience of the so-called KarMMa-2 study, where this was taken into a larger setting and really showed impressive clinical benefit for a much larger group of patients. In fact, the overall response rate was around 75%, and the median duration of response was approximately 11 months in this large phase 2 study, pointing to the fact that you can engender high-quality response that's durable, again, with the single approach. This requires no necessarily maintenance to follow or anything like it in these trials, simply just the approach. And these were, of course, in penta-refractory, highly pretreated patients. So the signal there very encouraging, and further studies are now ongoing to take this further into earlier settings with this approach with the BB2121 platform. Now, one of the challenges is a so-called cytokine release syndrome. And I want to emphasize this because it seems to occur more broadly with immune therapies. And obviously, there are various ways of grading this, but in its severest forms, it can be extremely challenging. The good news is that we've become much better able to understand it on the one hand, and at the same time treat it. And so this has fortunately become much less of a problem for the BB2121 platform that it may have initially raised itself as being early in the development of these platforms. So the good news there, generally manageable and relatively low rates. But we do realize that this is an important challenge going forward, and we're looking at another novel strategies to try and minimize this as a late complication. Now, in that same spirit, the cilta-cel platform, which is led by Janssen originally, the Legend platform from China is where this technology came, is incredibly powerful. It's again an autologous CAR T-cell, but basically it has 2 BCMA targeting sites, which gives it additional potency, as well as these 2 domains. And again, the signal derived from this particular approach has been particularly impressive, actually. And what's basically happened is we've been able show this to be extraordinarily active with a very high response rate, and very importantly, also associated with durable disease control. One of the challenges, however, has been that there is more toxicity associated with this particular approach. So this is an area of increasing focus and intense investigation to better understand how to minimize the toxicity and yet preserve the efficacy that's been seen. So again, the so-called CARTitude studies, KarMMa is for the BB2121 studies, CARTitude is for the cilta-cel platform, and these studies are ongoing. Now, I want to touch briefly that there are other platforms as well. I'm not going to be, in the interest of time, talking about every single one, just to give you a sort of smorgasbord of those that are currently under study. There's one by the CARSgen team, the so-called CT053 CAR, which is looking very excitingly at a similar targeted approach, but with additional nuances for patients with relapsed refractory disease. And again, preliminary results are very exciting from this approach. And I think what I rather like is the poetry of all of these studies. This is called the LumaCAR study, which is looking at this approach. And the initial response data are particularly attractive. As you can see, 94% of patients responding, including some very high quality responses seen. And this is obviously an exciting early signal. Now, if you look at the summary of the CAR T approaches overall, I've tried to summarize them on this slide, but what you can see here is that this has been taken on very broadly by a number of important platforms, and the early data are very promising. And what we're hoping to do is minimize the side effect profile and maximize the benefit. But as you can see, really from the construct of the duration of responses, they're encouraging at approximately a year. Median progression-free survivals are also good. But this raises then a critical question of how do you maintain responses with this approach? And so future directions include new antigens, new CARs, and we'll touch a little bit on this in a moment. Also, we need to be sure that with these CARs, there's an ability to rapidly manufacture, because one of the challenges has been to be able to move quickly to other platforms and deploy these platforms for our patients who obviously need them fast, not to be on waiting lists that last many months. And obviously, there are other strategies of combination therapies and maintaining remission. One particular target on BCMA I just want to touch on, just to especially acknowledge my colleague, Eric Smith, was at Memorial, and now we've been very lucky. He's come to us at Dana-Farber. He's a brilliant scientist. He's identified GPRC5D as a very important target in myeloma. And early data from this particular approach are particularly attractive. And then just on a lighter note, just to raise a smile, there's another CAR called a BAT CAR, no less, which is a binary activated T-cell with a chimeric antigen receptor. And this is under study and coming into clinical evaluation. And the simple point for sharing this is that these are a very exciting new direction for our patients with immune therapy, with some of the early results being, I think, quite unprecedented in terms of quality of response and depth of response. And what we're really hoping is that this will translate into real-world benefit for our patients with the eagerly anticipated FDA approval of ide-cel later this month. Now, beyond CAR Ts, what about bispecific T-cell engagers? These are so-called BiTEs. And basically, this is a different approach because it doesn't necessarily require the T cells to be taken out of the body and given back. This tries to do it all within you. And the very good news here is that, again, this has become a very exciting area of research. And I summarize here some of the critical studies that are currently ongoing. There are several in the mix. And I'm going to spend a little bit of time on each of the ones that are probably most mature. But what you need to see from all of these, and they range from a very large study in phase 1 of almost 150 patients strong to smaller studies of 50 to 40 patients or so. But these particular approaches are showing very high quality of responses, but at the same time also a manageable tolerability profile, recognizing that CRS is an important one to be on the lookout for. Now, again, not to blind you with the science, but simply to emphasize a point that actually Brian made earlier, that these are approaches that can be originally used in the hospital setting and then move quickly into the clinic thereafter, and are typically repeated every 2 to 4 weeks or so, depending on the nature of the particular BiTE platform. But what we're learning here is that by using these multi-targeted approaches in terms of bringing together both the BCMA target and the CD3 antibody, we're seeing dramatic responses in patients. So what I showed you just a moment ago was preclinical data to support this particular BiTE going forward. And this was lovely data presented by Dr. Rodriguez and colleagues at ASH. And what you can see here is that the overall response rate was 80% in doses that were considered active. And this was actually a Q3 dosing schedule with only 48 hours of inpatient requirement at the very beginning. So this is a very exciting platform. And similarly, when we look at other approaches, the data preclinically are extremely exciting. And in this context, again, this was actually lovely data presented by Dr. Garfel, who's very well known in the CAR T space as well, where he was able to show with this particular approach that he does a 73% response rate, including high quality responses. So yet another platform, again, showing benefit in this setting. Not to be outdone, the GPRC5D target, this is not BCMA, I want to stress to add, but GPRC5D, and this is very important because if BCMA has already been used, this target then becomes very important. This was excellent data, actually, that was preclinically summarized here, but clinically presented by, in fact, the Mount Sinai group by Achai Chari at ASH, in which Achai showed really nicely not only a favorable tolerability profile, but a response rate of around 70%, including some very high quality responses. So this was a true first-in-class off-the-shelf therapy targeting GPRC5D with ongoing expansions ongoing. And it's great to have Donna as part of the panel today because I know she was involved in this study. So in terms of BCMA targeted immunotherapy, I've just sought to sort of summarize it here by showing you that we've got obviously the CARs, we've got the bispecifics and the BiTEs, but at the same time, we also have the antibody drug conjugates. And you'll hear much more about this from Rafat in just a moment. But this is obviously a truly off-the-shelf approach in which we combine the BCMA targeting with the chemotherapeutic, methadone in this case, and the results have been quite remarkable. But I think what's particularly interesting is when this was combined with other therapies, such as pomalidomide, most recently at ASH, the data from Dr. Suzanne Trudel were extraordinary. And I know Rafat is going to come to that in a moment. You can see here how immunotherapy combined with other strategies is doing remarkable things. And this really brings me on to my next point, that as we think about BiTEs and CAR Ts, we will probably be bringing some of these new maintenance strategies, such as building on the IMF platform, into this space. What I mean by that is that these may be drugs that we add to the CARs, add to the BiTEs going forward. And probably the best or my best candidate in this respect is iberdomide. This is CC220, as it's called. It's in the CellMod class. And this may be an ideal platform that will follow CAR Ts and BiTEs in the future as a cellular therapy maintenance strategy, in my view. The reason I say that is because obviously all our patients will have had immunomodulatory drugs such as lenalidomide and pomalidomide early. And so after these immunotherapeutic approaches, strategies aimed at targeting to maintain the immune response may include agents such as iberdomide. Now, the big question about these cell mods is, in fact, can they do it even on their own? I mean, do they need, in every case, a CAR T or something innovative like that up front? Well, clearly, our data to date would say no, that in fact, these cell mods have really been a next step up. This is data from our own study. This is the 92480 platform. And just to give you some background as to what these cell mods do, they are so-called E3 ligase modulators, and they're in a different fact. They're related to the IMiDs, but they're different. They're that much further distant in terms of their pharmacology and their construct, but their effects are dramatic on the cereblon E3 ligase complex with this extraordinarily powerful targeting of ALOS and Icaros, which in turn not only has tumoricidal effects, but critically profound immunostimulatory effects that are way beyond what we've hitherto seen. And as a result of that, we've been very pleased to see, and this is just from single agent 480, 55% of our patients in this phase 2, or rather RP2D dose cohort, recognizing the number here is relatively small, but the signal is nonetheless very strong, that just with 480 plus dexamethasone in these highly refractory patients, we saw a 55% response rate. Now, important to remember, this is just a pill, taken 3 weeks on, 1 week off, and the steroids administered weekly. So this is a highly convenient approach for going into a whole new area in terms of response. Now, these patients, the 55% responding patients, were refractory to everything that was available up to that point. They were triple-class refractory, and so therefore constituted an exquisitely vulnerable population. So we're very excited by that. This is one of the patients in this particular study. I'm very grateful to Dr. Nizar Balis for sharing this particular PET CT, but this patient in particular had extramedullary disease in their liver, which is obviously a very ominous finding. And as you can see, with 480, cycle 3, day 1, this large mass had completely disappeared. This is just a pill, and exploiting the immune system to so do. So really a very, very exciting approach, which we think is particularly promising. So I just wanted to close with these summary points and just perhaps some future directions. Obviously, as we think about the relapsed refractory space, and we're going to hear beautifully in a minute from Rafat about the various options that we have, whilst we fully recognize that triplet therapy is the gold standard at the moment, to which things can be added, we're very hopeful that this new wave of immune therapies, in particular targeting BCMA, be it the antibody drug conjugates or the CAR Ts to come hopefully very soon with the approval at the end of this month, and then perhaps the real impact of the so-called bispecific T-cell engager therapies can really take our treatments up to a new level for our patients. And by that same token, will the cell mods provide us with that same next big push? And so I'd like to close by saying, as we think about this and move these strategies earlier, will we be in a position to offer patients earlier in the course of their disease truly immune-targeted approaches that generate durable responses that continue for many, many, many years, and provide patients even with the opportunity for treatment-free periods where the immune system has done such a good job that essentially they can be off therapy and then enjoyed a true treatment-free period. And you might say, does that mean functional cure? I think that that's a possibility indeed. I'm very cautious about using that term because myeloma over so many years of caring for my patients always humbles me. But at the end of the day, I do think this is one of the most exciting and promising horizons for all of us as we look into 2021. I want to thank you very much for your kind attention. And obviously, Brian, open for any questions. Thank you.
Thank you so much, Paul, for this comprehensive overview of the immune therapies, especially the CAR T and then the bispecifics. Very, very exciting. And we have questions coming in here already. But